Okay, so continuing where we left off with translation, we're now going to talk about translation in terms of what happens biologically, and this will explain why you start at a start codon and why you end with the stop codon, which we haven't gotten to either. And then we'll have a kind of practice problem we might end with so that next week we can talk about what happens when things mutate. So translation is going to require all of the RNA varieties. So we've been mostly focused on mRNA, the messenger RNA, but last class I talked about two other kinds of RNA, tRNA and rRNA. rRNA actually makes up the ribosomes, and we're going to talk about what a ribosome is. tRNA is transfer RNA, and it is a folded RNA that interacts with mRNA and also interacts with amino acids. So here's kind of what a tRNA looks like. It's got two ends to it that are important. Really, how it folds and what it looks like in the middle is not vitally important. The two ends are important, and there are different varieties of tRNA. There are about 20 varieties of tRNA, one for each different amino acid, and they have different anticodons. There actually are more than 20, but we're going to kind of pretend there are only 20. Some of them actually have some flexibility to them, so there end up being enough to cover every of those different codons. So on one end is something called an anticodon. It's the region of the tRNA that is going to bind to the mRNA. So this tRNA has an anticodon UAC. So what three letters of mRNA do you think this would bind to? Right, but mRNA, not DNA. But you're very close. So how would you change that to make that RNA? AUG, right? Yeah. So AUG. So the mRNA, AUG, is going to bind with the tRNA UAC. So this would be the anti-codon, and this is the codon. So an mRNA codon, why did I write two T's there? A-N-T-I, there we go. Anticodon. So we have an anticodon on the tRNA, we have a codon on the mRNA, and each tRNA will have a specific amino acid, and anyone have their chart out in front of them or just know off the top of their head what the codon AUG codes for? AUG is the start codon. Yeah, methionine. So this would be met here. So this amino acid, methionine, is on a tRNA that has an anticodon UAC on it. There are lots of other tRNAs. They have different anticodons. They have different amino acids. And you could figure that out based on that chart, and we'll do some problems based on that again. So again, the tRNA anticodon UAC is going to bind to an mRNA codon there, and the amino acid binding site here binds to the corresponding amino acid here. So it has an amino acid on one side, anticodon on the other side. And this is how this molecule here, tRNA, is actually going to decode the message in mRNA. It's going to be able to read it and bring the appropriate amino acid with it. So your cells don't have a little table. They aren't looking up the different codes and putting the right amino acid. What they use is tRNA that has a particular anticodon and brings the appropriate amino acid. Okay. So the location where the tRNA and the mRNA meet up is the ribosome. And this is what a ribosome looks like. So a ribosome is built from RNA and proteins. So it's rRNA, ribosomal RNA, mixed with proteins. It catalyzes the assembly of amino acids into a protein. So what does that tell us a ribosome is? If a ribosome is something that catalyzes a reaction, it's an enzyme. Yeah, so a ribosome is an enzyme. It's an enzyme made out of mRNA mostly and proteins. So it's actually one of the few enzymes that is not entirely protein. It has a large subunit and a small subunit, and we're going to learn the large subunit has some binding sites on it. The small subunit binds to the mRNA. They're going to assemble around the mRNA to allow for the process of translation to occur. This is what they'd look like if we actually showed the amino acids, the RNA, and the protein that is mixed together. So the green part here is rRNA, ribosomal RNA, and it's folded intricately like this. You can see lots and lots of folds to it. And the purple parts are proteins. You can see it's mostly mRNA, 5,000 RNA bases, but it also has 49 proteins in it. It's a mixture of the two. In no way do you need to remember the exact makeup of a ribosome. But you will need to know the function of it as we go along. Okay, so ribosomes are going to be the sites of translation. This is where mRNA and tRNA will meet up. Ribosomes are actually they're like the factory building. They're going to provide the correct locations for these things to come together and really guide them into the right locations. And we're going to see that this actually provides these little binding sites that expose 3 mRNA bases at a time, which allows the tRNA to nicely come in and bind in there. And it catalyzes the assembly of the protein. Okay, so here's kind of back to an overview of everything that goes on during protein synthesis. So transcription followed by RNA processing occurs in the nucleus. What we're talking about now, translation, is happening out in the cytoplasm, either bound to the ER or free in the cytoplasm, where the ribosome meets up with the mRNA and a protein is produced. Translation, just like transcription, is going to occur in the same 3 steps: initiation, elongation, and termination. But this time we are initiating the production of a protein, we are elongating the protein, and then we are terminating the production of the protein. And all 3 of these steps are going to happen at ribosomes. So translation, similar to transcription, 3 steps. And I'm going to highlight all 3 of these steps. Any idea where initiation is going to occur, where on an mRNA initiation will occur? Well, it'll be an interaction between an anticodon and a codon, but what will be the codon? Remember from the very first question today? Yeah, it'll be AUG. It'll be the start codon. So, exactly. Okay, so initiation occurs. So the mRNA that's been processed, has gone out into the cytoplasm, left the nucleus, and the first thing that happens is it meets up with the small ribosomal subunit. So this thing up here is the small ribosomal subunit. Small ribosomal subunit will bind to the mRNA, and it will kind of run along the mRNA until it reaches AUG. So anything on the mRNA that is prior to that AUG, so 5' of the AUG, is ignored by the ribosome. Ribosome looks for that start codon to know where to start making a protein. And if you remember from Wednesday, a lot of this stuff that's prior to the start codon, which is called the 5' UTR, 5' untranslated region, that has other functions. It might be a localization signal to tell the mRNA where to go, where in the cytoplasm does it end up. So there's a lot of things that happen here, but they have nothing to do with the protein that's being made. The first thing that relates to the protein is AUG, and so that's what the small subunit looks for. Once the small subunit has attached to the mRNA, the large ribosomal subunit will bind, as well as the first tRNA molecule. So tRNA that has the anticodon UAC and has methionine is going to bind to that start codon, and the ribosome assembles around it. That's initiation. So anything prior to AUG is ignored, and AUG is always the very first codon that is recognized by a ribosome. So again, if I ever give you a question like that first one again, the first thing you're going to do is look for AUG, and you're going to ignore everything before it. Okay? All right, so the tRNA, remember, is going to be complementary to the base pairs in the mRNA. So the tRNA anticodon UAC matches up with the RNA codon AUG. And the tRNA, again, is carrying the amino acid methionine. This is going to set up something we call the reading frame. So because the ribosome has sat down here at AUG, this determines the first codon, and the next three letters are going to be the second codon. So until that point, there was no determination of where those triplets lie. And those triplets could have been here, here, here. They could have been anywhere here. So this determines the sets of three triplets. And we basically know that now every three bases from here on out is going to be read. So until you find that start codon, you don't know where your reading frame or where those triplets line up. If AUG had been somewhere else here, it might have changed the reading frame. And if you look closely at this graphic, you might notice that there is actually a second AUG here. So right here is another AUG. So there actually could be a second protein made from this same mRNA if it had started here rather than here. And you'll see that it's a slightly different reading frame. They actually they don't line up exactly. And we'll talk about that a little more as we go forward. Right. Okay, so this sets up the reading frame. And as the ribosome sits here, it's going to have two binding sites, the first of which is filled with that first tRNA that was bound to the start codon. The second binding site is now open for a tRNA to enter, and different tRNAs will enter until the tRNA with the correct anticodon for the next triplet of bases enters that binding site. Second tRNA will enter the binding site, and if it matches, the amino acids that those two tRNAs are carrying will be covalently bonded. So this next codon, GGA, meets up with the anticodon CCU, and that tRNA brings with it glycine, GLY. So now that these two are bound to the mRNA, a reaction is going to occur that forms a covalent bond or a peptide bond between these two neighboring amino acids. And we're getting the start of elongation, which is making our protein. So what do you think is going to happen after this? Yeah, so there's going to be, you're going to go to this third site, but what does the ribosome have to do? Yeah, it's going to have to move over. So the ribosome is going to shift down that direction, and this site's going to be available, and we're going to see that this tRNA actually will leave. It'll break off. So the ribosome shifted down one. This tRNA will separate, and it'll actually break away from the amino acid. And there'll be a new binding site open for the next tRNA to enter and bring with it another amino acid. It's going to covalently bond, and it's going to continue to happen. The ribosome will shift down. The previous tRNA will leave. It'll break off, and a new tRNA will enter. So the first tRNA leaves, the ribosome moves to the right, and the third tRNA comes in. And this will continue to happen over and over and over again. Okay, this is called elongation. Okay. Notice that although you keep adding new tRNAs, the initial amino acid remains connected here, and you now have these linked amino acids. So you're going to continue to link to add new amino acids, and they're always going to be attached to one of the tRNAs that's still in the ribosome. And as the ribosome shifts down, it just kicks out the tRNA without the amino acid. This tRNA will actually be recycled. There are enzymes that will add amino acids to tRNAs. So as you digest proteins, or as proteins break down, your own proteins break down in your lysosomes, you are freeing up amino acids. Those amino acids could get tacked on to these empty tRNAs. So a methionine will be added to this tRNA, and it can go and participate in protein synthesis again. So this process will continue occurring as the protein grows. Okay. What do you think is going to stop it? Okay, eventually it's going to reach one of those stop codons. So one of those regions that didn't code for an amino acid, but coded for stop. When that happens, rather than a tRNA, so there's no tRNA that matches up to UAA, UAG, or UGA, which means there's no tRNA with the anticodons AUU, AUC, or what is it, ACU. So these anticodons do not exist. So those three anticodons do not exist. So there's nothing, no tRNA with an amino acid that would bind to UAA because there's no AUU anticodon. So when UAA is in the ribosome, something else will bind to it called a release factor. The release factor protein binds to the stop codon, and this causes a change in the reaction that is occurring. So no longer will there be any covalent bond formed here. Instead, this is going to cleave this final bond and will free up the functional protein. So this protein is the polypeptide that has been formed will be released at that point. So stop codon is reached. The release factor will bind rather than a tRNA, and this causes the protein polypeptide to detach from the mRNA and the tRNA that it was attached to and free it up. It will now fold based on its polarity and become the functional protein. Okay. So that is termination. Okay, so, all right. So we've seen initiation of transcription occurs when the start codon is reached, so AUG is reached. The ribosome assembles around the mRNA. The tRNAs start entering the binding sites one by one, matching up to the codon, adding and bringing in their appropriate amino acids. The amino acids will link together in the ribosomes, and the ribosome will keep shifting down one. That's the elongation of the protein. This continues to happen, matching up the codons with the appropriate tRNAs until you reach one of the stop codons, and at that point, the release factor enters and terminates translation. And you have your polypeptide, and finally, the polypeptide detaches and folds, and you have a functional protein. And this was just the last step on the last slide. Okay. Mm-hmm. All RNAs will leave the nucleus eventually, but mRNA, what makes it special is that it actually has the code for making the protein. So the mRNA here is the strand on the top. So this right there is mRNA. So it has to leave the nucleus. tRNA and rRNA, when they leave the nucleus, the tRNA becomes this structure, which has the anticodon and attaches to amino acids. And the rRNA becomes this structure, the ribosome. So all of them function outside of the nucleus. The mRNA is the recipe. The tRNA is kind of the decoder of the three, and the rRNA is the enzyme. Right. The polypeptide is the protein. So the rRNA is the ribosome. This green one is the tRNA, and this light green one up at the top is the mRNA. Yep.
So do the tRNAs have protection from the destructive enzymes outside the nucleus as well, or just the messenger?
It's a good question. Probably not, but they get manufactured pretty regularly. Okay. So once they break down, so if they get chewed at either end, the two ends of the tRNA are the amino acid binding site and the anticodon. So if either of those ends gets destroyed, it's no longer functioning. So it would just be broken down by a lysosome and remade. So your cells are constantly making tRNAs, are also constantly making rRNA, because ribosome would have the same thing. It would be degraded eventually. Yeah.
And so the ribosome actually moves down the messenger RNA?
Yes.
The messenger RNA doesn't move through the ribosome? Or is it?
Well, you could say either way. They're moving relative to each other.
Okay. Well, I
Yeah.
didn't know if one was stationary and
They're,
past it.
well, only when they're attached to the rough ER, the ribosome really is stationary. So I guess in that case you would say the mRNA is moving relative to it. But when they're out in the cytoplasm, who knows? They're kind of, yeah. So some texts will say the mRNA shifts, some will say the ribosome shifts. Regardless, they're moving relative to each other. Okay, any other questions about this? So basically understand how this works. So what is actually reading the message on the mRNA, like most directly? Yeah, the tRNA. So the tRNA anticodon is reading the message, and because of the enzymes that add the appropriate amino acid to the appropriate tRNA, the tRNA brings in the right amino acid. So really this didn't show this, but when a binding site opens up, lots of tRNAs will kind of go in. Most of them won't fit, and they'll only stick if they have the right anticodon, and then they'll have the reaction occur. So it's a lot of trial and error that's going on, just things bumping into each other, and then eventually the right one fits in. Yeah, so if the wrong one goes in and fits, and then you can get a mutation to your protein. Yep. Sure. Or if for some reason the tRNA had been mutated and it put the wrong amino acid onto the wrong tRNA, that would also lead to mutation. This is stuff we'll get into really on Monday, but yeah, there's a lot of chances for errors here. And errors happen all the time. All the time, yeah. Most of them are caught, and we'll talk about that a little more on Monday. Okay. Yeah. So, oops. So I have a little video, which I'll show you, that is basically shows this process. So let's take a look at that.
This student is cramming for her biology test with only a frosting-covered donut for lunch. Specialized cells in her pancreas respond to the increasing amount of sugar in her blood by releasing insulin, a small protein that regulates blood sugar levels. Let's go inside one of these cells to see how this protein is manufactured. The instructions for making insulin are coded by a segment of DNA in the nucleus. In transcription, an enzyme zips along the DNA, forming RNA, shown here in red. RNA nucleotides line up with their complementary DNA partners, transcribing the information in DNA into RNA. As the RNA grows, it is processed in several ways. First, a modified guanine nucleotide is added to the beginning as a cap. Also, segments of the RNA strand that do not actually code for the protein are removed, and the remaining segments of RNA are reconnected. Finally, extra adenine nucleotides are added to the end of the RNA strand, forming a tail. The completed messenger RNA, mRNA, now leaves the nucleus. The message in mRNA is translated into a protein in the cytoplasm. First, a transfer RNA, tRNA, arrives, carrying a specific amino acid. The small subunit of a ribosome attaches to the mRNA. Now, the large subunit of the ribosome attaches. A second tRNA docks, bringing another amino acid. The ribosome helps to form a covalent bond between the two amino acids. The mRNA shifts, and the first tRNA leaves. A new tRNA brings another amino acid. The ribosome helps to form a new bond, and the process is repeated. Notice that one end of a tRNA molecule has a set of 3 bases called an anticodon that pairs with complementary bases on the mRNA. The other end of the tRNA carries a specific amino acid. Different types of tRNAs carry different amino acids. In this way, the message in mRNA is translated into a specific sequence of amino acids. For proteins that will be secreted from the cell, like insulin, the ribosome docks on the rough ER, and the protein grows into the ER compartment. The new protein molecules are packaged in a vesicle that is transported to the Golgi apparatus, where many proteins are processed. However, insulin is packaged in a vesicle that leaves the Golgi and is then processed. Proteins secreted from the cell are shipped to the plasma membrane. Here, insulin is secreted from the pancreas and begins to regulate the student's rising blood sugar levels.
All right. Yeah, so the processing we haven't talked about yet. We might get to that a little bit later, but so hopefully that gives you an idea. That's the general process of transcription and translation. All right, questions on that? Does that sound pretty straightforward? So translation really is all about the ribosome being a location for the mRNA and the tRNA to meet up. The tRNA is going to bring the appropriate amino acid to the ribosome and match up codon with anticodon in order to bring the right amino acid in to make the right protein. And as the ribosome works its way down the mRNA, you're going to get a longer and longer polypeptide. Just a few more things on this. One, this is not happening one at a time. It's not like one mRNA and one ribosome are going to function. As soon as that start codon is exposed again, as soon as the ribosome has shifted down enough for the start codon to be exposed, another ribosome could assemble around it and the protein could be produced again. So most mRNAs are fairly long. They're going to have many, many, many ribosomes on them at the same time. So you're going to be making multiple proteins simultaneously. Ribosomes as well are not specific to certain proteins. So basically they are machinery that will make any protein. So whatever mRNA is present in your cell is going to be made into a protein. As long as it has a start codon and a stop codon, it's going to make a protein. And as soon as those degrading enzymes chew up the mRNA enough to destroy the coding region of the protein, it's going to start making at first just a mutated protein that is truncated, and then eventually they'll get rid of the start codon and it won't make any protein anymore. So that's kind of that extended poly A tail is going to lengthen the amount of time that it's making a functional protein. Okay, as the protein is built, it will fold based on the polarity of its amino acid subunits, and that kind of was shown at the end of that video. So this protein, actually, as soon as it emerges, it already starts folding based on the polarity. So if this was polar and this was polar, they would form a bond between them. It would pull them closer together, and the protein would start to fold like that. Okay? Yeah, well, yeah, we're going to talk about that once we get to the regulation section of this chapter. We'll talk about how DNA is packaged. So, but you're right, it is packaged as well. Okay, I'm going to have you do this. We'll do it again on Monday, but I just wanted to pull up this chart to make another point. This is a table that we've made through experiment of mRNA codons. So if you were looking at mRNA, one of the big mistakes some students will make is they'll say, oh, it needs to be converted into the anticodon, into tRNA, before I can do anything about it. Yes, that's how your cells do it. Your cells use tRNA to read the mRNA. But we have this cheat sheet that is built directly off of mRNA codons, so we need that to read them. So I want you to spend a few minutes now with this problem. Here is a strand of DNA. I want you to tell me the protein that this DNA codes for. All right. Most of you? Anybody still? Most of you seem like you're done. So what's the first step? What do you have to do first in this to answer that question? Yeah, convert it to RNA, which is a process called transcription. So it'll make it into mRNA. So that would be UAA, UG, AAA, CAU, CGA, GUUU, AA, CGAU. Right? Good job. So then I need to go from mRNA to protein. And this will be translation. Right. So do I start with UAA and say, oh, it stopped, so there's no protein made? No. So I need to look for AUG. This is going to set up my reading frame. So now I know where my triplets are. You could designate these if you'd like. It's nice. And so AUG would be methionine. Then the next triplet would be AAA, which is going to be lysine. And then CAU, which is here, histine. And CGA, which is arginine. And then GUU, which is valine. And then UAA. All right, so what happens there? Stops. All right, so is stop an amino acid? No. So you don't need to write anything. You just stop. So this would be the protein. Right? Met, lys, his, arg, val. Is that what everyone got? All right. Fantastic. Good job. Okay, so we're going to stop here. And we'll do a problem similar to this at the start, and then we'll actually come back to this, and we're going to talk about mutation on Monday. So keep those notes. I'll probably have you do the same exact problem on Monday at one point, because it's already on my board. So, all right. Have a good weekend.